All right, looks like we're ready to go, everyone. Hello, welcome. My name is Gabriel Benton, Senior Scientist here at Trevigen Incorporated. And I'm here today to present the webinar entitled The Next Step in the Evolution of 3D Culture, Utilizing Extracellular Matrix to Enhance Multicellular Spheroid Models for Proliferation and Invasion. Okay, and over the course of this webinar, first I want to present some background and then go over our tools and reagents, specifically our Spheroid Proliferation and Viability Assays and our Spheroid Invasion Assay. And then I'll wrap up with a summary and a question and answer session. Okay, so first for our background. Okay, I just want to start off with a timeline of events which are important for most cell-based models and specifically our spheroid model. I hope everyone can see this all right. So the origins of cell culture date back almost 130 years to 1885 when Wilhelm Rauch's first cultured embryonic chick cells. Then in 1907, Ross Harrison first utilized the hanging drop to culture embryonic frog neural tubes. This same model was used many years later to establish spheroid cultures, so it's relevant to what we're talking about here. It's essentially a drop that hangs from an inverted microscope slide or plate where the cells can only adhere to each other. This allows the cells to self-organize without the interference of an adhesive surface. Later on in 1951, George Gay established the first immortalized cell line, HeLis. With the establishment of this cancer line, it kind of started the ball rolling for standard models for cell culture to become commonplace in laboratories everywhere. In 1970, Robert Sutherland began using spheroids as tumor models. He found that spheroids of comparable size mimicked the physiology of small avascular tumors. These multicellular tumor spheroids provided heterogeneous cell populations, some of which exhibited drug and radiation resistance. The only problem was that the model was limited because it did not work with all cell lines. Next, in 1985, Hinda Kleiman invented basement membrane extract, also known as Matrigel. This is one of the most commonly used reagents for organotypic cell models. It's extracted from the Engelbreth Holmes swarm tumor, which is propagated in mice. And this extract provides a full complement of soluble and insoluble factors for the tumor or tissue microenvironment. And it is also relevant to our model because we use a lot of these tissue extracts here. Then in 1991, Makoto Kawada demonstrated that spheroids have an extracellular matrix and that the extracellular matrix is necessary for spheroid formation. In 1998, Trevigen introduced the Cultrex extracellular matrix proteins, thus beginning our journey into 3D culture and the development of assays related to extracellular matrix. Then in 2006, Andrea Vasquez used extracellular matrix proteins to induce spheroid formation for cells that were previously unable to form spheroids, demonstrating that exogenous extracellular matrix was sufficient to drive this process. Also in 2006, Henry Hirschberg utilized spheroids for extracellular matrix invasion, proposing a new tumor invasion model to recreate early events in metastasis. So this timeline indicates how long we've been working on these models. It's been steadily evolving over the last 130 years. And these are some critical discoveries that have brought us to where we are today. So I think it's first, we're getting ready to talk about spheroids and about 3D culture. It's first to be good to establish what 3D culture is. Basically, 3D culture, they're in vitro cell culture models where cells can grow and assemble in 3 dimensions. As we can see here at the bottom of the page, we have our 2D culture, which is just basically a flat monolayer. And we can envision this growing in X and Y dimensions, whereas In 3D culture, we have what is represented by a sphere here is cells being able to grow in X, Y, and Z coordinates. So they're able to form, take the shape of the tissue of origin for each cell type. And they assume similar morphology or structure as well as function, gene expression and protein expression. And the key to creating 3D culture really is to be able to kind of recreate the in vivo environments and allow the cells to initiate their own cell program. Okay, and then it's also important to look at why we use 3D culture. So why is it important? As I discussed on the previous slide, it's more physiologically predictive than 2D. We're able to actually grow organotypic structures rather than just sheets of cells. It's faster than animal models, more species relevant than animal models because we can use human cells. It's less expensive than animal models. We have more control over our experiments than we do in animal models because we're able to control what cell types and what different molecules or treatments are made within the cell culture device. There's less regulation than animal models, and 3D culture is also compatible with animal models. Some of these, for some models, structures are grown up in 3D and then they can be implanted into animals to improve the efficacy of the model. And as we see here, kind of below, is just it's embryonic mouse salivary gland. On the left is just the gland by itself at the beginning of the 3D culture, and then what we see is during the course of the 3D culture in a laminin-rich extracellular matrix, we see both ductal and kind of acinar structure development, similar to what we see within the mouse. Okay, so the purpose really of 3D culture and everything we're talking about here with spheroids, it all relates back to being able to create physiologically relevant models. And on the left here, I have a slide that represents mammary tissue. And really what I just want you to see here is that it is composed of many different cell types and different ECM, and it's a very complex environment. And in our assays, we use tissue extracts. So these are proteins that are extracted from the tissues from animals, and this provides a better or more biologically active matrix for these cells to grow and invade into. On the right, we see the model of cancer progression, where we start with a normal epithelia, which progresses down to hyperplasia. We have kind of hyperproliferative response in the cells, and they become a little crowded, which progresses to dysplasia, where we see abnormal cell types. When these abnormal cell types start to proliferate into a primary tumor, it's progression into the carcinoma in situ. And then from carcinoma in situ, it will progress to metastasis, where the cells will invade out of the primary tumor and into the surrounding ECM through the basement membrane, through the connective tissue, enter the vasculature through intravasation, migrate to foreign sites, extravasate out of the vessel and form a secondary tumor. And metastasis is really the critical point in cancer. It's really the most lethal step of the cancer process. So it's important to be able to have good models for looking at this. Our spheroid models, the spheroid proliferation and viability model, it recreates the primary tumor, such as you see in the carcinoma in situ. Then when we look at the spheroid invasion model, it's reconstructing these very early steps in metastasis where the cells are invading out of the primary tumor and into the surrounding ECM. Okay, this next slide represents basically just different cell models for the same cell line. These are MDA, MB231 breast cancer cells. The first figure A, is 2D culture, where the cells form a monolayer. See kind of a cobblestone morphology. Figure B is a traditional 2D culture, where the cells are basically in a single cell suspension. They're added to a basement membrane hydrogel, and what we see are several small invading structures within the culture. Figure C is our tumor spheroid model, and we see one spheroid within the plate. Okay, and what's important is the size. The spheroid needs to be large enough to where it can recreate some of these physiological properties associated with tumors. And I'll talk about that on the next slide. We see in figure D that when we embed this spheroid into an extracellular matrix hydrogel, the cells can invade out of the spheroid and into the surrounding matrix, similar to what we see with the early metastasis. Okay, and so this is an Engelbrecht Holmes swarm tumor. It's a tumor that we process here on a regular basis. Just wanted to show, kind of illustrate how we look at 2D, and obviously the single cells, the sheets of cells, they're not representative of what we see in a tumor. Whereas when we look to the spheroid model, we see large cell aggregates. We can recreate these physiological gradients that are associated with tumors. Basically, what happens is the spheroid is surrounded by the cell culture media, which contains the nutrients, growth factors, oxygen, everything that the cells need to grow. And within the spheroid, we have multiple layers of cells. And each layer of cells represents somewhat of a barrier, a diffusion barrier for these nutrients to diffuse into the spheroids, as we can see with this, the top arrow. And at the same time, within the spheroid, within the core of the spheroid, these cells are producing catabolites. They're producing waste products. It's a low pH, very hypoxic environment, and we have the same problem where everything that's produced within the core of the spheroid has to diffuse out, and that's this bottom arrow. It's just showing how these are diffusing out, and as a result, we get heterogeneous cell populations where the cells in the core, in this very toxic environment, actually become necrotic or apoptotic. You see cell death. Whereas the cells in kind of the intermediate zones, they don't have sufficient nutrients for cell division, but they do have sufficient nutrients for viability. They become quiescent. And the cells that are on the kind of the outer surface, having full access to these nutrients and everything else, become proliferative. And we can, and this here very well illustrates this process and how we get these heterogeneous cell populations. This next slide. Here we have U87MG, which are glioblastoma, astrocytoma cell line, and PC3s, which are prostate cancer cell line. And we have different numbers of cells that are seated in each well, and this is at the top left-hand corner. So in this top figure, it says 391. The next figure is 781, and then 1563. So we're seeing an increasing number of cells, and as we increase the number of cells in each well, we're increasing the size of each spheroid. Okay, and also what we see is that if we look back at the 391 here in the U87MGs, that we see very little cell death with these smaller spheroids. And then as the cell number increases and the spheroids get larger, we see increasing amounts of cell death within the core of these spheroids. And this shows us how we have this kind of this diffusion zone where the cells are able to get sufficient nutrients to maintain viability. And I think I forgot to mention that what we used here was a calcium AM was our viability reagent. And so what happens is the cells internalize the calcium AM, and it is metabolically converted to calcine, which fluoresces and is trapped within the cells. And the red staining that we see in our dead cells is aethinium bromide. In dead cells, a lot of times the plasma membrane becomes compromised, allowing the aethinium bromide to diffuse into the cell and intercalate the DNA. Whereas with viable cells, the aethinium bromide cannot diffuse in through a intact plasma membrane. So for our tools and reagents, first look at our 3D spheroid proliferation and viability. I wanted to start out by looking at sample preparation, specifically looking at how we set this assay up. It's something that we've had some questions before in the past couple of weeks, and I think it's worth spending some time on, so that if you do set this up in your lab, it will make the transition much easier. So first we'd start out just calculating volumes for our 10x spheroid formation ECM, our complete growth media. So this is growth media used in cell culture, includes your fetal bovine serum and any other media additives that are needed for your cell of choice, and the cells at 1 million cells per mil. What we see here, what I've set up here is for one well, and this can be scaled up for multiple wells. And for one well, we're looking at 50 microliter volume. We have a 10x spheroid formation ECM. So if it's 10x, we'll use 5 microliters. For the cells, I actually have entered a value of x, and this can be variable. And this is an important point to mention, that different cell lines have a different size, and generally what we're looking to do is create a spheroid of a specific size, so between 400 and 600 microns in diameter. This is where we see a lot of these physiological gradients created. So this may require a little optimization based on your cell model. If we go over to the next column, where we have 3,000 cells per well, we see that it would be 3 microliters would be X. This is a good starting point. We've used this for several different cell lines, and we're generally we're within that 400 to 600 micron diameter range. And then the remaining 42 microliters would be the complete growth media to make up the 50 microliter volume. Okay, so now we can talk about how we actually mix things together. We start off thawing our 10x spheroid formation ECM on ice at 4 degrees Celsius overnight. So we want to pack it on ice and in the refrigerator. These extracellular matrix proteins will they'll polymerize at temperatures between 15 and 20 degrees Celsius. So we want to keep them cold. We want to keep them liquid while we're setting up the assay. If they do polymerize, then we will not be able to use them to set up the assay. So it's very important to keep them cold. If we're not going to use all of them in one shot, we can divide the 10x spheroid ECM into working aliquots and store it minus 80 degrees Celsius. But avoid freeze thaws. That's why we say working aliquots. And we also want to keep the reagent on ice throughout the procedure. And now that we're ready to dilute, we'll add our cold complete growth media to the tube, then add our 10x spheroid ECM and pipette up and down to create a homogeneous solution. Okay, now we're ready to add our cells. And so just a quick note on cell culture. We always recommend culturing your cells to about 80% confluence and then dividing them as recommended by the manufacturer or the supplier of your cells. Basically, having healthy and actively growing cells is very important for most cell-based assays, and it's good to practice uniform tissue culture for these assays to get the best reproducibility. So we'll harvest the cells, centrifuge and aspirate off the media, and resuspend the cells at 1 million cells per mil in complete growth media. And then transfer the calculated amount of cells to the spheroid formation ECM, and again just pipette up and down a few times to create a single cell suspension. And once the 10x spheroid ECM is diluted, it's stable and it doesn't need to be on ice anymore. So once it's diluted, it's now stable. We can work with it in the hood without ice, without any problems. And so we take our cells in the spheroid ECM. We add 50 microliters to the 96-well spheroid formation plate, centrifuge at 200 times G for 3 minutes in a swinging bucket rotor. And what this does is it pulls all the cells to the bottom of the well and puts them all in close proximity, and this helps to facilitate the spheroid formation process, and it also helps to accelerate it too, so it helps both with the time and quality of the spheroids. We also recommend the swinging bucket rotor specifically because that pulls the cells to the bottom, the very bottom of the well, which also gives us kind of the best placement for viewing. Okay, and then then we take these plates and insert them into the cell culture incubator for 72 hours. And over that time, the cells will assemble into spheroids. And then we'll prepare a 2x treatment, a solution containing 2x treatment compounds in complete growth media. And we use 2x because we have 50 microliters of spheroid formation ECM in the well, and we're going to add 50 microliters of treatment. So after that addition, it'll become a 1x treatment. After we've added the treatment, we incubate at 37 degrees in the tissue culture incubator for a desired treatment period. We're generally looking at 72 to 96 hours, but these spheroids can remain viable for a long time in culture, as we'll see in a later slide. Okay, and this slide is just somewhat of a summary with a timeline for spheroid formation. You see it takes 3 days for spheroid formation. This is a conservative value. Some lines will form spheroids faster. we find that we just keep this at a constant for all the lines we work with, and we have not had any problems. And then we have a 3 to 6 days of spheroid proliferation. And we can use image analysis to evaluate the growth of these spheroids. And that basically just means taking a picture and using image analysis software to look at growth rates. As we see here, we have our MDA MB231s looking at 3D spheroid growth. We started with 3,000 cells per well. This was a 35-day assay. In the pictures on the bottom left-hand corner, okay, in the upper left-hand corner of each picture is a number, and that number corresponds to the days in post-spheroid formation. And what we see is that these cells will, the spheroids will actually continue to grow for 20 days. They'll reach a certain size. For these cells, it was a little over 1 millimeter in diameter. And then once they hit this certain threshold, the spheroid cannot grow any larger. However, the cells in the spheroid remain viable. And this has been described all the way back to the 1970s with the early spheroid models, and was once proposed as a model to look at tumor dormancy. And it's related to not having vasculature to further support growth of the spheroid. If we look at the figure on the right, this just demonstrates the importance of the spheroid formation ECM. Figure A are MDA MB231s in the absence of the spheroid formation ECM. They just form these very loose colonies. And in B, we have with the spheroid formation ECM a nice compact spheroid formation. Okay, and we can also look at spheroid growth over time, as well as inhibition of growth using this technique as well. On the left, we see spheroids. This measures the spheroid area. Right below the graph are pictures of the spheroids themselves, and we see that the spheroids expand over time, over this 4-day assay. And we also see nice kind of tight error bars with these measurements as well. And that's mainly because since we're only having one spheroid per well, we can control the size of these spheroids pretty exactly. And you see that we get a nice growth curve just by analyzing these images. The figure on the right, we're using bleomycin, which is a chemotherapeutic agent. We're treating the spheroids for 96 hours after spheroid formation. And we see a decrease in spheroid growth associated with this treatment of bleomycin. So the growth measurements, they're very informative. However, they don't tell us really much about the actual cell proliferation or the cell viability. It can be misleading if you have a large aggregate of dead cells and you're measuring. You cannot get very specific data. So kind of to accommodate this, we've added fluorometric and colorimetric cell viability reagents in these kits for endpoint analysis. Okay, so this first slide that's loading up, it's a fluorometric quantitation with Resazurin. Resazurin. All right, and then we use the exact same spheroid formation protocol and treatment protocol as we do for the image analysis. The only difference is at the very end of the assay, we add 1/10 volume of Resazurin. And the cells will reduce the Resazurin to Resarufen, which is fluorescent. And we use this to quantify the cell number over a 1 to 4 hour period. Okay, and as we see here, here is our data. On the left is a standard curve based on different cell seating densities for the spheroids. So we have different cell densities, different spheroid sizes, and different, actually an increase in our relative fluorescent units associated with an increase in cell number, as we'd expect. And then on the right, we're treating with bleomycin, and we see a decrease in the cell viability associated with increasing bleomycin concentration, as we would expect, giving us an inhibition curve. Okay. Similarly, for colorimetric quantitation, we use MTT. Again, we're using the exact same protocol for spheroid formation and spheroid treatment. And then at the end, we're just adding 1/10 volume of our MTT reagent, which the cells will reduce to a purple formazan crystal. And then we will actually solubilize the cells and the crystals and read absorbance values. And this is important because we've looked at several of the other colorimetric methods where the cells produce a soluble product like MTS and WST, and we've seen a lot more noise, a lot more variability in the readings. And we believe what's happening is that these spheroids are of sufficient size that they're interfering with the absorbance readings through the wells. So using this MTT assay and actually solubilizing the cells with these formazan crystals is giving us the best, most reproducible data possible. Okay, and so here is our graphs. Again, we have a graph with varying numbers of cells seated in each well, forming different size spheroids and corresponding to an increase in absorbance. And then on the right, we have an inhibition curve where increasing amounts of bleomycin decreases the absorbance, thus the cell viability for these MDA MB231 cells, spheroids. And then we've also used this assay. This was done using the fluorescence assay to look at 2D versus spheroid viability with staurosporin treatment. And what we saw was that the 2D cells were much more sensitive to the staurosporin. They had an IC50 of around 0.1 nanomolar, whereas the spheroid cells and spheroids, again these are the MDA MB231s, had a IC50 closer to 50 nanomolar. And this is what's been shown in the past is that staurosporin is that cells in hypoxic environments become resistant to staurosporin treatment. And obviously cells within these spheroid layers do exhibit hypoxic conditions, and this is at least a part of the mechanism here of how the spheroid cells are resistant to the staurosporin treatment in comparison to the 2D cells. Okay, and on the next slide, still coming up here. I don't think you see it. Here it is. Okay, so here we're just showing that our extracellular matrix proteins do induce spheroid formation in multiple low adhesion models. So in our low adhesion plates, we see that with no spheroid ECM, there is no spheroid formation. This is the top row. Whereas when we add the spheroid formation ECM, we see over time the first panel is 0, the next one is day 1, day 2, and day 3. We see compact spheroid formation. Similarly, with the hanging drop, which I mentioned before, it dates all the way back to 1907. We see that in the absence of the spheroid ECM, these are for MDA MB231 cells, there is no spheroid formation, and then when we add the ECM proteins, we do get spheroid formation over the 3-day period. Now the low adhesion plates do provide us some advantage here in that we are able to do some centrifugation to help, like I said, get the cells close in proximity and help kind of accelerate the process, as you can see between the two models. And here is just a slide. We're looking at day 3 and day 7, these MDA MB231s with the spheroid formation ECM, and we see similar spheroids of similar size and morphology for the 3 and 7-day incubations. Again, we prefer the low adhesion plates. They're just they're easier to work with and a little bit easier for imaging as well. All right, this next slide, we're looking at a cell line that does not require extracellular matrix for spheroid formation. So they spontaneously form spheroids. These are U87MG cells. As we can see below in the pictures below the graphs, they do not need the ECM to form spheroids. However, what we've found is that the addition of the ECM actually appears to accelerate spheroid growth. So it's providing support for these cells to help accelerate the spheroid formation process, which could be advantageous for these models. Okay, now we're moving on to spheroid invasion. Sorry, it's just taking this slides a minute to load up. And for the spheroid invasion, we're going to follow the exact same process for spheroid formation as we did with the proliferation and viability assays. The only difference is that we're going to be, after the spheroids are formed, we're going to be incorporating an invasion matrix. So, as we can see here, during spheroid formation, after about 48 hours of spheroid formation, you want to make sure and thaw the invasion matrix on ice at 4 degrees Celsius overnight. Again, this is very important because these ECM proteins will polymerize at temperatures between 15 and 20 degrees Celsius, so they need to be kept cold. And also to make sure you keep it on ice during the full process when working with it. So as soon as the invasion matrix is thawed out, we'll take the spheroid formation plate with the spheroids in it and put it on ice at 4 degrees Celsius, so in the refrigerator, for 5 minutes. This will quickly chill down the plate and the spheroid formation ECM and the spheroids themselves. And we'll add 50 microliters of cold invasion matrix to the pre-chilled spheroid formation plate. Okay, and then we'll transfer these to a centrifuge. So it's pre-chilled down to 4C, and centrifuge 300 times G for 5 minutes in a swinging bucket rotor. And what this does, first of all, keeping everything cold gives the spheroid formation ECM and the invasion matrix time to fully mix, become homogeneous. Also, the centrifugation helps to embed these spheroids and place them in the center towards the bottom of the well, where they're best placed for viewing and analysis. So once these spheroids are centrifuged down, set the plates at 37 degrees Celsius in the tissue culture incubator for 1 hour, and that will polymerize the invasion matrix, fully embedding the spheroid. Then we'll add 100 microliters of complete media, or media containing chemoattractant such as FBS, with the 2X invasion modulating agents. So this is where you could add inhibitors. And it's, again, a 2X concentration because we're diluting it out. The 100 microliters of media will be going on to an existing 100 microliter hydrogel. And we keep these, we want to make sure this is warmed up to 37 degrees Celsius, this solution, to prevent depolymerization of the matrix. Then we will incubate at 37 degrees Celsius, 5% CO2, incubate the plates for a desired invasion period. We generally incubate between 48 and 96 hours, and then analyze the plates for invasion. But you can analyze the plates real time and get photographs as it's happening also. And as we see here, just another timeline with a kind of a summary of the assay. We have 3 days for spheroid formation, and then 2 to 4 days for spheroid invasion, giving a total assay time of 5 to 7 days. And then for analysis, we use image analysis, but there are some choices here in terms of how this analysis is conducted. You can use bright field microscopy or fluorescence. We've used red fluorescent protein expressing cells, as well as cells that have been labeled with lipophilic membrane dyes, such as PKH67 and PKH26. And they all seem to work really well in the system. The advantage of the fluorescence, of course, is that you have a better signal to noise. It's much easier to pick up the structures. Whereas with the bright field, the advantage there is it's label free. So here's a quick graph showing this invasion assay. We have the MCF7s, which are a non-invasive cell line, and our MDA MB231s, which are invasive. The top rows, okay, these are conducted over 4 days. Column 1 is day 1, column 2 is day 2, column 3 is day 3, column 4 is day 4. The top row for each sample is no matrix, so there's no invasion, no hydrogel for the cells to invade into. And the bottom row is with invasion matrix. For the MCF7s, we see virtually no invasion of the cells out of the spheroids. For the MDA231, there's no invasion for no matrix. We do see the spheroids grow a little bit. But when we add the invasion matrix, we see a very robust invasive response where the cells are invading out of the spheroid. And we can quantitate this as such. Here we see MCF7, no difference between no matrix and invasion matrix, no invasion. With the MDA MB231s, we see a subtle increase in the size for no matrix of the spheroid size, whereas with the invasion matrix, we see a very robust response, very robust increase in size. All right, and our next slide just want to show that there are different cell lines exhibit different invasive potentials and morphologies. As we can see here, we have prostate, PC3, prostate cancer, MBA, MB231s, which are breast cancer, HT1080s, which are human fibrosarcoma, and U87MG, which are glioblastoma, astrocytoma. We can see, like with the PC3s, we do not have the same level of response as we see with the other cell lines. We also see that, like with the U87MGs, that the core seems to expand more so than it does in the other cell lines. However, we get kind of these more spindly invasions coming off. The invasion in front doesn't seem quite as thick as it does with the other cell lines. So this just kind of shows us some different invasive potentials and morphologies that are associated with different cell lines. Next, we'll see an example of invasion inhibition using a known invasion inhibiting compound, sulforaphane. If we look at the pictures, you can see with the increasing concentration of sulforaphane that we see a decrease in these protrusions coming out of the spheroid. And if we look up at the graph, we see a dose response associated with this decrease in cell invasion. And again, these are for the MB231 cell line. Next, we see a Boyden chamber versus a spheroid invasion. So the Boyden chamber is in the blue, and the spheroid invasion is in the red. We see that the Boyden chamber has a much higher IC50 for etoposide, which is a known chemotherapeutic agent, whereas the spheroid invasion has a much lower IC50, approximately 5 micromolar. And this is mainly because the etoposide is a topoisomerase inhibitor. It requires replication for its pharmacological activity. We see that the Boyden assay is very fast. It's generally about 24 hours. The cells really do not replicate very much over this period of time, and that has less of effect, whereas in the spheroid invasion model, the cells, this is a 96-hour assay, cells are going through multiple rounds of replication, and the compound is able to exert its activity more robustly. And so that shows you this assay could be better suited for looking at compounds that affect that are effective during cell replication or extended incubation periods. Okay, next we're just looking at a known treatment of proliferating spheroids with a known anti-proliferative agents. We have 2 millimolar hydroxyurea and 0.5 micromolar triapine. You see, this is, I apologize, the bottom of the figures got messed up when we uploaded them, but it's day 0 on the left and day 4 on the right. And we see virtually no growth over the 4-day period with these inhibitory compounds. When we use these compounds in the invasion assay, again, over a 96-hour period, column 1 is 0, and column 2 is 4 days. We see that the cells still have, although there is inhibition, there's still a pretty robust invasive response in the absence of proliferation. So, in summary, 3D cultures provide suitable environments for cells to self-assemble into organotypic structures mimicking the morphology and function of their tissues of origin. Spheroids provide ideal tumor models based on morphology, size, physiological gradients, and the formation of heterogeneous cell populations. Spheroids exhibit an extracellular matrix, which is instrumental in the self-assembly of cancer cells. The addition of ECM proteins promotes spheroid formation and growth, and ECM hydrogels provide physiological scaffolds for cancer cells to invade out of tumor spheroids, mimicking early events in metastasis. And here's just a summary of our tools we have available for 3D culture: spheroid proliferation, / viability, and invasion. And I've included, also included a set of references here. This goes back to our timeline, as well as there's a few references here at the end which aren't included in the timeline, but they're just very good, good reading material on spheroids, more recent papers and reviews. All right. Thank you all very much for your attention. I'd be glad to take any questions. Okay. So here's a question: do fibroblasts form spheroids? This is actually a very good question. We have not experimented with fibroblasts and spheroids. We have seen cold cultures using cancer cells with fibroblasts to make spheroids. This is definitely something that we would want to investigate. There may already be literature on this out there. But it's a very good question. And we'll look into that and see if we can answer that for you. Okay, here's another question. Can fully formed spheroids be collected or removed with their structure intact? So yeah, we've been able to pull the spheroids out and actually do. Usually we fix them first and pull them out and embed them in paraffin and do sectioning. So they can be harvested. These are low adhesion environments, so the spheroids aren't actually attaching to the plate. They're more or less free floating, so they're very easy to harvest. Okay, we have a question here. Have you tested embryonic stem cells? At this point, we have not, but this is something that we're very interested in doing. We actually have plans to look at embryonic, or not actually embryonic, but to look at stem cells in these assays. Okay, another question is, can the spheroid invasion assay be used for endothelial cell-based spheroid angiogenesis assays? And so, yeah, this assay, based on what we know from our other angiogenesis assays, should be fully compatible with endothelial cells. We just need to complete the qualification so that we can officially make this claim. But there's no reason why this assay should not work with endothelial cells. Okay. Do you need to digest the extracellular matrix to harvest the spheroids in the ECM? The answer is no. The concentration of the ECM proteins is low enough that the spheroids are free-floating. You don't need to digest the matrix, at least not for the spheroids if you're just growing them for spheroid proliferation / viability. Once they're embedded into the matrix, you actually would want to fix and pull the entire plug out for analysis, because you want to look at the cells that have invaded out of the spheroid as well. Okay, we have a question regarding hypoxias. As you mentioned that there is hypoxic conditions in the center of the spheroid. How do you measure this? I believe there's been two ways that this has been done. It's been done several times in the literature. I believe there's a product called hypoxia probe. It's a substrate that the spheroids can actually be treated with prior to fixing, sectioning the spheroids and looking at the gradients. Some people also will look at markers such as hypoxia-inducing factor 1, HIF1 as well, and just basically do immunostaining. You'll harvest the spheroids, or fix them, harvest them, section them, and look for differences. Okay. Well, it looks like we're pretty much out of time here. We can we'll try to answer any of these unanswered questions through email. We'll get back to you. If you have any other questions, I have my contact information is on the first slide, my email address. Just please feel free to email me, and we can take it from there. Thank you all very much for attending. Have a good day.